So we shall start our class. We share the screen. Class will join us. No problem. So we were our topic known as genetics. We were on genetics. So and by now we have to know how to cross using the crossing and use of the Punnett square. So come on, we join genetics that the crossing and the Punnett square. Hizi back zote ni just crossing tu, hakuna kitu ingine. We are almost done the topic. So ile kitu muhimu sana ni just the crossing and the crossing. Can you mute yourself? So just the crossing and also use of the Punnett square and umuhimu. Hizi zingine unapewa tu. So codominance, tulikuwa kwa codominance. We say that codominance is a condition where alleles or genes are both dominant and they both express themselves phenotypically. Not genotypically, phenotypically. Phenotypically and I think I already explained the difference between phenotype and genotype. In simple term, I say that phenotype is the observable characteristics ambayo inawezaona. Naweza sema this person is tall, is short. So the examples of the codominance ni hizi nne. ABO blood groups. Blood groups sasa ni blood group A, blood group O, blood group A, blood group B. Because blood groups are only these three. Blood groups ni hizi tu tatu. So inaweza kuwa either A, inaweza kuwa B, inaweza kuwa O, ama inaweza kuwa AB. So the blood groups huwa zinatoka to those three letters. Kuna mahali zingine zinatoka. And I think it is sickle cell anemic. Sickle cell anemia is the cell that has doesn't have the red blood cells. Also white and red flower colors. Also white and black coat of the guinea pigs. Easy to tatu inakwanga. For example, look at this. Swali wanasema when a pure breeding black guinea pigs were crossed with pure breeding with white guinea pigs, so tunaona black guinea pigs with white guinea pigs. The F1 offspring had a coat with black and white patches. So all of them they express themselves. So iko na black na iko na white. Using letter G, ndio nakupatia letter. Using letter G to represent the gene for black coat, and letter H for white color, work out the genotypic ratio of the F2. Ujue ni F2 si F1. So huwezi fanya F2 kabla hujapata nini? Huwezi fanya F2 kabla hujapata F1. So lazima upate F1. F1 is the first generation. So the F1 sasa itakuwa G for black, si ndivyo? So G and H inakuwa ina fuse. Na already hizi rules zake nimekwambia. So nimekwambia sasa E1 inaenda na hii, still hii inaenda na hii. Tumejaa hiyo moja. Tunakuja hii, tunafuse side na hii pia tunafuse side. So itakuwaje? Itakuwa GH will be GH. So this will be GH. So this will be GH. This also will be GH. Hii pia itakuwa GH. Hii pia itakuwa GH. So all of these is gonna black and white. All of them. So this is known as F1 generation. So the first generation unaita nini? F1 generation. So saizi swali imetuliza nini? The ratio of F2. So F2 sasa ni we self this. So tunachukua GH na hii ndio umefanya sawa. So the GH and GH. So kama kawaida G inaenda hizi itakuwa GG, then itakuwa GH, then hii itakuwa GH, hii pia itakuwa HH. So this ratio ndo sasa inaita nini? Genotypic ratio. Ni 1GG, 2GH because hii ni na hii pia ni GH, 2GH and 1HH. And that's known as genotypic ratio. Kusema phenotypic ni all of them one in black, huyu ni black, na this is known as black white. Na swali ndio umetuambia. Swali umetuambia nini? Umetuambia The F1 offspring had a coat with black and white. So this is F1. F1 zote hizo zinaitwa nini? Black and white. Zinaitwa black white. So ni two black white and one white. So as long as you understand how to fuse them, hizi hii ingine ni rahisi sana. Ingine rahisi. Na certain plant species which is normally green. So the plant species ambayo inakuwa normally green, a recessive gene for color N causes the plant to be white in color. So recessive gene tumesema is a gene that does not express themselves. They does not show themselves. So this causes the plant to be white in color. Such plant die at an early stage. In the heterozygous state, the plant are pale green in color but grow to maturity. Give a reason for the early death of the plant with the homozygous recessive. Wachana hizo mambo zote ya homozygous recessive. Swali tu ni kitu moja tu. Na unafaa uelewe tu kitu moja. Species inafaa ikuwe ngapi? Ikuwe color gani? Si ni green. Imekuwa ngapi color? Imekuwa nini? Imekuwa white. So plant kuwa green inasaidia nini? Green inasaidia ikuwe photosynthesize. Since the green color imeenda, hakuna chlorophyll. Unajua the green color ni chlorophyll tu. Since hakuna chlorophyll na chlorophyll zimeenda, then inamaanisha there's no manufacture of food. Na kama ya plant haiwezi jitengenezea chakula yake, inafanya nini? Kama hakuna chakula, the death will follow. So hii story yote wewe wachana, usitake kujua. Just know that plant ingefaa ikuwe mbeleni, ingefaa ikuwe nini? Green, si ndivyo? Na saa hizi ni white. So wanasema tu the color. So green na hii ni white, saa hii imefanywa imekuwa white. So what do the plant lack? It lack the chlorophyll, which is the green matter. So the leaf do not have the chlorophyll, does not manufacture the food or not carry out the photosynthesis. There's no development of the tissue. Na hii inaleta nini? Death. So if normal plant were crossed with the pale green plant, so the normal plant ambayo ilikuwa na N. So kama recessive gene ni N, recessive gene ni small n, the dominant gene itakuwaje? Itakuwa capital N. Because nilikwambia last night that the capital letter inaonyesha dominant. The small letter inaonyesha recessive gene, the one that will be outmasked. So if a normal green plant was crossed, normal green ambayo iko na N sasa, iko NN, which is crossed with the pale green. So pale green ni color gani sasa? Pale green ina contain nini? Look at the question. Sasa such plant die at an early stage. In heterozygous state sasa. Heterozygous ujui. So heterozygous itakuwaje? Heterozygous, jana ni usiku nimekwambia, is someone that have dominant and recessive at the same time. So iko na capital letter na small letter. Simple term. So this is known as heterozygous. Na jana tulitumia nini? Capital T small t. Capital T small t. Heterozygous. Na jana nimetumia 3 examples. Sijui kama unakumbuka. The 3 example was, there's only, for example, the toneless. There's only 3 things. This ambayo inaonyesha toneless, this ambayo inaitwa shortness, and this is the another one ambayo inaitwa nini? Heterozygous store. Nimekwambia anything ambayo iko na the same letter inaitwa homozygous. Inaitwa nini? Homozygous. Anything ambayo iko na different letter inaitwa nini? Heterozygous. Inaitwa nini? Heterozygous. Hivyo unafaa uelewe. So what is happening? If the normal green plant ambayo ilikuwa na NN is crossed with the pale green, which is N, ambayo ilikuwa heterozygous, umetuambia already kwa question, what is the genotype for the first filial or the first generation? First filial generation, F1 generation. Unaita first filial generation. So you just cross kama kawaida. So the moment, sasa tumeona trick genetics ni moja tu. The moment you understand these letters, gani homozygous, gani heterozygous inakaaje, gani inakuwa dominant, recessive genes zinakaaje. The moment you understand those words, that's all about the genetics by the way. Hakuna kitu kingine. Anything that they will be tested in exam, especially in paper 2, itakuwa tu the definition of terms. Wanakuambia nini? Define codominance, define heterozygous, define homozygous, define alleles. Hizo words ambayo na watakuuliza tu definition. Hii zingine ya crossing, the moment you know the genetics. Almost 5 marks, out of 3 marks. So this I think there's no need of repeating myself. So unarudia hivi, then it will be like like this. So itakuwa nini? Itakuwa two green and two pale. Two green and two pale. So sometimes we may identify the unknown genes, unknown genotype, unknown genotype. Genotype ambayo haijulikani. So there's some as we can identify them through three things. Number one is test cross, back cross, and self-pollination. Self-pollination ni ile inaitwa selfing, we just self it. Test cross ni, this is a genetic cross between individuals of unknown genotype. We just cross using unknown genotype, showing the dominant characteristic with the recessive individuals. Back cross ni is a genetic cross where an offspring is crossed with one of the parent. So back cross sasa ni unatumia offspring, you just use the offspring, that's the kids ama the watoto, the offspring, then you cross with one of the parent, then unatafuta nini another parent. Lakini still hizi zote ni ile tu iko na jina tofauti, lakini inaleta tu ile common crossing tu, inaleta tu the common crossing, hazina tofauti. Self crossing ni ile ya genetic of cross between members of the same filial generation. For example, first generation no una cross. Tukuja kwa test cross. Test cross for example ni kama hii sasa, for heterozygous dominant. We cross using nini? Heterozygous dominant. Na heterozygous nimekuambia nini? Heterozygous ni hii, this is known as heterozygous and this is homozygous. So heterozygous you cross with homozygous and you get the product. That's known as test cross. Test cross for homozygous. Homozygous ni ile you cross the homozygous and homozygous. For example, TT, capital TT, we cross with small TT, and then you just cross the normal way. Then selfing of homozygous. Also, this is a TT, homozygous dominant. All of them ni homozygous. Just cross it. Also, this is known as selfing of heterozygous dominant. Isotin heterozygous plus heterozygous. So they just give the same thing. Hii hakuna kitu muhimu sana. So nikuje kwa inheritance of the blood group. ABO blood groups. So blood groups, there are two antigens by the way. From two work tukurudia, there are two antigens. Kuna antigen A and antigen B. Antigen A and antigen B. So the inheritance of the blood group is determined by three alleles. Kuna allele A, allele B, and allele O. Allele A, allele B, and allele O. Meaning that allele A iko na antigen A, allele B iko na antigen B, and allele O haina antigen. So they prevent formation of antigen A and B on the red blood cells. Tulikuwa tunasema hivyo wakati tulikuwa form 2, the first topic tulikuwa tunasema blood group A iko na antigen A. Yani there's two antigens, antigen A and antigen B. Now blood group O, they have no antigen. So allele A and allele B show codominance to each other, while allele O is recessive to both allele A and allele B. So allele A and allele B, they show codominance. Codominance tumesema nini? Codominance ni they are available, they are present, and all of them they express themselves. Hakuna moja inafichwa. Lakini allele O inafichwa. Na nani anaficha allele O? Allele O inafichwa na either A ama inafichwa na either B. Inafichwa na either O ama inafichwa na either B. Either O ama B. So this is the result tunaweza pata. So tunaweza pata either genotype inaweza kuwa A ama A. So this ina contain antigen A. It's found under the blood group, the phenotype blood group A. This is the blood group A. So anything ambayo ina contain A, inakuwa nini? Blood group O. Mbona tunasema ni blood group A? Because tumesema O is recessive. Na tumesema recessive ni nini? Recessive ni kitu inafichwa. So blood group A ina contain antigen A. The genotype ambayo tunaweza pata from the blood group A ni mbili. Either ni AA or AO. Blood group B pia iko na antigen B kama kawaida. The genotype ambayo tunaweza pata ni BB or BO. And the blood group AB, the genotype ambayo tunaweza pata ni B. And the antigen it contain A and B. And the blood group OO tunaweza pata genotype OO and the antigen are none. Hakuna antigen. That's all. Hivyo unafaa uelewe tu. So hizi nyingine sasa, as long as umeelewa the blood group A unaweza pata either genotype ambayo iko na AA na AO, na mbona tunapata AO ama BO? Kwa sababu gani? Tumesema O is recessive to A and B, is recessive to A and B, yani inamaanisha inafichwa. So hapa iko na O, lakini phenotypically tunasema nini? Phenotypically ni kitu tunaona characteristics of that person. Phenotypically tunasema aje? Tunasema this person is blood group A and the person is the blood group B. Kwa sababu gani? Because O is recessive gene. Recessive gene is a genes ambayo inafichwa. Genes ambayo inafichwa. So ukielewa hizo zingine just need to normal crossing. So for example, angalia. A woman with blood group A, married a man with blood group B. So a woman akona blood group A and a man akona blood group B. The first born had blood group O. Give the genotype of the parent. So tuseme hata kama mwanamke akona blood group A, the genotype inaweza kuwa mbili. Either inaweza kuwa AA, tuseme either inaweza kuwa AA or AO. Kama ni BB inaweza kuwa BB or BO. So lakini swali sasa imeendelea na wamekwambia their firstborn had a blood group O, akona blood group O. So meaning that kama firstborn akona blood group O, kama ni pure AA and pure BB, there's no way watapata blood group O. There's no way firstborn yao itakuwa nini? Atakuwa O. So meaning that their genotype of the parent itakuwa AO and BO, and that's how you determine. So the mother ni AO, the father is BO. Just tunatumia tu akili yako. Use the Punnett square to work off. Punnett square tumesema ile tunaweka box. So tunaweka father tunaweka hapo juu BO, then mother tunaweka hapo chini AO. So tuna times 2. A times B itakuwa AB, A times O itakuwa AO, B times O itakuwa BO, then O times O itakuwa OO. So and this is the reason why they got it. Explain how codominance is illustrated in the inheritance above. Codominance imeillustratiwa kiaje? Codominance ni ile inakwambia nini? They show themselves phenotypically. For example, hapa they have blood group AB. So naona the blood group AB already naona hapa. So the reason is gene A and B are both expressed phenotypically to antigen A and B on the red blood cells, then they thus forming blood group AB. That's known as codominance. Codominance nimekuambia ni ile both of them they are available and they show exactly the same thing. Yani haionyeshi any color because jana tulipitia kitu ambayo inaitwa for example, wacha nikwambie ya this thing tulipitia jana. Nikuonyesha tukirudi klasi ya jana kidogo. Yeah, this part, yeah, known as incomplete dominance. Tulipitia kwa incomplete dominance. So tofauti ya incomplete dominance na codominance ni nimekuambia these are three things unafaa uelewe: complete dominance, incomplete dominance and codominance. Nimekuambia nini? Complete dominance, incomplete dominance and codominance. Incomplete dominance ni ile moja na fichwa. For example, the TT, ile T kubwa na T ndogo. Saizi hii ni recessive, sio? Inafichwa kabisa na haionyeshwi. So na hii sasa tunasema nini? Tunaita this is a tone. Inafichwa hii T ndogo. The shortness inafichwa. And that's known as complete dominance. Incomplete dominance ni ile vitu mbili ambaye zikimate. Inakupatia another color. For example, black and white they can meet and inaweza kupatia nini? For example, kitu kama yellow. Inaweza kupatia nini? Kitu kama yellow. So they give you another color. And that's known as incomplete dominance. Codominance ni ile zote zinajionyesha. For example, this iko na color black na huyu pia iko na color black. After they met, they give you black white. So hakuna color tofauti imeformiwa. The color ambayo imeformiwa ni black white. Hakuna color tofauti. That's known as codominance. I think you understood that. I think umeelewa hiyo. So that's the difference between complete, incomplete and codominance. So angalia this person, Mrs. Romanas claimed that her child was wrongly identified at hospital, so that she took away the wrong baby. So both Romanas and her husband are blood group A. So both of them are blood group A. The child, the child blood group is O. Romanas justify in her claim, make a genetic cross using Punnett square to explain your answer. So come up, both of them they are blood group A. Both of them they are blood group A, meaning that they have blood group AO and AO. Since their kid akona O. So you just multiply like sasa itakuwa AA itakuwa AA times O itakuwa AO, times O itakuwa AO, O times O itakuwa OO. So from the justifies, from the cross, it is possible that the child could have blood group O, blood group O. Therefore, the woman is not justified as the parent could be heterozygous, because tusema blood group A and blood group A, the blood group B iko na homozygous and heterozygous. Homozygous ni wakati iko AA BB inaitwa homozygous BB. Homozygous. That's homozygosity. Also, there is a possibility that they can be heterozygous. That is AO. AO. This O is recessive ambayo inafichwa. AO. AO. So that's how you do it. So blood transfusion ni ile sasa tunasema nini? Tunasema antibodies ina cause antibody. So the human serum or human blood ina contain kitu inaitwa nini? Antibodies. These antibodies react with blood group antigens in the blood group cell membrane. So the antibodies and antigen present in people of different blood group are shown below. So for example, sasa hizi hapa tunaingia opposite. Ni ile tu ya form 2. So ile tu ya form 2. Tunasema kama mtu ni blood group A ako na antibody B. Kama mtu ako na blood group B ako na antibody A. Kama mtu ako na blood group AB hana antibody. Kama mtu hana antigen ambaye ni blood group O ako na antibody A and B. Hii ni kazi ya form 2. First topic form 2. So the antibodies and antigen are different, are opposite. Antibodies and antigen. Let's know that antibodies and antigen are opposite. So kama mtu akona antigen A ama blood group A akona antibody B. Kama mtu akona blood group B ama antigen B akona antibody A. Kama mtu akona blood group AB na akona antigen A and B ana antibody. Kama mtu ana antigen akona antibody A and B. Yeah? B. So something that's known as agglutination. Agglutination ni clumping of the blood ambayo zinakutana na zina clot, zinashikana yani, when the recipient antibodies correspond to the antigen of the donor's red blood cells. So thus, for example, if someone akona blood group A, mtu kama akona blood group A akona antibody B, cannot receive blood from a donor of blood group B. Blood group B ambaye akona antigen B. Because there are something that's known as antigen antibody reaction. Antigen antibody reaction, they causes clumping of the blood, and clumping of the blood is known as agglutination. So meaning that mtu ambaye anapatia damu, blood transfusion ile tu kupatiana damu, mtu ambaye anapatia damu lazima antibody yangu na antigen yangu na antibody yangu na antigen yake zisikuwe the same. Those antibodies and antigen ikikuwa the same, the blood clumping happen. For example, do I mean? For example, let me use example from this from my group. For example, what do I use? Mulki. Or let me use that person because we iltire said to me Mulki. For example, tuseme Mulki akona blood group A. Then iltire akona blood group B. Blood group B. So Mulki akona blood group A sio? So akona antigen A. Akona antigen A na antibody B. Antigen A na antibody B. Huyu pia akona antigen B, antibody A. Meaning that the antibody of this iltire the antibody of this L3 is the same as the antigen of Mulki. Antigen of Mulki. As long as these antigen and antibodies are the same, hakuna blood transfusion. Na in case blood yenu ukifanyiwa blood transfusion, blood agglutination ina happen. Blood agglutination ina happen. Agglutination nimekwambia ni clumping of the blood. Blood zinashikana. And kama blood imeshikana na hakuna haiwezi tembea, na lead nini? Na lead death. Na lead death. That's how it happens. That's how it happens. So tukuje kwa inheritance of the rhesus factor. Hii kazi ya form 2 tu. Hii ni kazi ya form 2, si kitu mingi. Rhesus factor is also another antigen found in the surface of the red blood cells. So rhesus factor also is another antigen ambayo inaformiwa ina contain in the red blood cells. So the dominant allele RH, rhesus inaandikwa hivi, determine from the formation of rhesus antigen known as antigen D. So rhesus inaitwa antigen D. Rhesus ukisikia tu ni antigen D tu in the red blood cells. So the recessive alleles ina contain RH. Yani RH inaonyesha ni small r. Kama the dominant inakaa capital R, the recessive inakuwa nini? Small. So nimekwambia hivyo. Nimekwambia dominant always is presented as capital letter, while the recessive is presented as small letter. Small letter. So the possible answer from this ni kitu tatu tu. That is either dominant, zote zikuwe dominant. Zote zikuwe dominant. Dominant inamaanisha nini? Ni RH ambayo ni RH. Yani kama total RH. Ama zikuwe nini? Ama zikuwe recessive zote RH zote small r. Ama zikuwe heterozygous, hizi ni homozygous sasa. Ama zikuwe heterozygous ambayo ni R kubwa H na R ndogo H. R ndogo nini? H. And this is known as heterozygous. Example 1. So wacha tukuja hapo also kwa negative and positive. So if the genotype is RH, the genotype ni E capitals, ama ni heterozygous, the rhesus antigen is found on the surface of the red blood cells, phenotype rhesus is positive. So tunasema ni positive sasa. So hizi mbili ambazo ziko na capital, anything ambayo ziko na capital R, tunasema is positive. Tunasema nini? Positive. Na anything ambayo iko na small r, zote RH sio. These small r ambayo tumesema ni recessive, tunasema aje? Tunasema ni rhesus negative. Rhesus negative. So question. A rhesus factor is controlled as the dominant gene dominated by RH. Heterozygous RH positive. Heterozygous ujue. So kuambia heterozygous ni R kubwa H ndogo, R kubwa H ndogo. Ujue hizi si letters tofauti. These are rhesus moja ambayo unaweka circle moja. So also these are rhesus nyingine unaweka circle moja. So these are rhesus. So these are heterozygous. These are heterozygous man married a rhesus negative woman. Rhesus negative woman inakaaje? Inakaa hivi. So akona RH zote ni small. So after that unaweka circle yake pia na hii pia unaweka circle yake, and then you just self it kama kawaida. Hizi ndio unafaa uelewe tu, hizo just self it. So solution ni hivi, unaona? So these are heterozygous and these are homozygous. These are negative. So after that you get rhesus positive and rhesus negative mbili. Homozygous rhesus positive. Homozygous rhesus positive kuambia aje? Kuambia ni RH zote. R kubwa RH because it's homozygous. Marries a rhesus negative woman. Rhesus negative woman ni RH R ndogo H ndogo and R ndogo H ndogo because it's negative. Then after that you cross it. You just cross it. After that you just cross it. So inshallah next night tutaanza nini? Tutaanza sex determination in humans. So this sex determination tutaangalia tu mambo ya chromosome. That's a man and a woman because tunasema female contain XX chromosome. That's a homogametic. Tunasema homogametic. And the male contain XY. That's a heterogametic. And that would be our next lesson, God willing.